Welcome to a new presentation in our Spring 2016 Cytology course at Virginia State University. My name is Hediye Labujamis and my fellow presenters are Fedwa Al-Jud, Noella Muma, and Nikia Lawrence. We are presenting a research article by Wrong and colleagues titled "The BH4 domain of BCL2 inhibits ER calcium release in apoptosis by binding the regulatory and coupling domain of the IP3 receptor." The BH4 domain distinguishes the pro-apoptotic BCL2 family members from the anti-apoptotic members. Its exact function is not fully understood. Thus, the research aims to investigate the anti-apoptotic role of the BH4 domain of BCL2, which is an anti-apoptotic member of the BCL2 family. Understanding its role will contribute to the overall understanding of the BCL2 protein, which may be used as a potential therapeutic target for diseases with accelerated apoptosis, such as Alzheimer's disease. BH4 binds to the inositol 145 triphosphate receptor, the IP3R, which is located on the endoplasmic reticulum membrane. BH4 binding inhibits IP3-dependent channel opening, which thus inhibits or prevents calcium from being released from the endoplasmic reticulum, thus in turn inhibit the calcium-mediated apoptosis. In the absence of a trophic signal, which is a cell survival signal, there is a caspase cascade activation. In normal healthy cells, the cytochrome C is localized between the inner and outer mitochondrial membrane. But in cells undergoing apoptosis, cytochrome C is released into the cytosol. In the absence of a trophic factor, BAD, which is a soluble pro-apoptotic protein, binds to BCL2 and BCLXL. BCL2 and BCLXL are anti-apoptotic proteins which are embedded in the mitochondrial membrane. BAD binding prevents BCL2 and BCLXL from interacting with the pro-apoptotic protein, which is embedded in the mitochondrial membrane, BAX, BAX, and then BAX is able to form homodimers, which permits the ion influx from the cytosol into the mitochondria. It is unknown why the ion influx causes the cytochrome C to be released into the cytosol, but that's the result. Cytochrome C is released into the cytosol and binds with APAF1, which is the apoptotic peptidase activating factor 1. APAF1 then activates the pro-caspase 9 into its active form, the caspase 9, which in turn, the caspase 9 activates the pro-caspase 3 into its active form, the caspase 3. This caspase cascade activation leads to apoptosis. In contrast with the presence of a trophic signal, there is inhibition of the caspase cascade. When a trophic signal binds, it activates the PI3 kinase, which is short for phosphoinositide 3 kinase. PI3 kinase then activates the AKT, which is a downstream effector of the signaling cascade. AKT is also called protein kinase B. AKT then phosphorylates the pro-apoptotic protein BAD, which is BAD. This phosphorylated BAD then forms a complex with the phosphoserine binding protein 1433. With a BAD sequestered in the cytosol, the anti-apoptotic BCL2 and BCLXL can then interact with BAX and prevent its oligomerization. With BAX activity is inhibited, the ion influx is inhibited. Thus, the cytochrome release into the cytosol is inhibited, which in turn inhibits the cascade activation.
The role of calcium in apoptosis. Calcium can induce apoptosis by mitochondrial/AR crosstalk. IP3 receptors on the ER are in close proximity with the voltage-dependent anion, thus facilitating transfer of calcium from the ER lumen to the mitochondrial matrix, leading to mitochondrial calcium overload and apoptosis. When the cell gets exposed to an apoptotic signal, the pro-apoptotic BCL2 BAD becomes dephosphorylated and binds to BCL2. This binding to BCL2 opens up IP, IP3 receptors on the ER, leading to the efflux of calcium out of the ER and into the mitochondria through the voltage-dependent anion channels. The influx of calcium into the mitochondria leads to the release of cytochrome C from the mitochondria. This cytochrome C then binds to the IP3 receptor of adjacent ERs, thereby enhancing calcium release from the ER, which then leads to the mitochondrial calcium overload and the release of more cytochrome C from all mitochondria. The excess cytochrome C in the cytoplasm then induces the formation of the apoptosome, in which capspaces are activated and thus apoptosis. The BCL2 family is a critical checkpoint in the intrinsic pathway of apoptosis. They are divided into two main groups: the pro-apoptotic proteins, which promote apoptosis, and the anti-apoptotic proteins, which inhibit or prevent apoptosis. The BCL2 family members are made up of at least one conserved region known as the BCL2 homology domain: BH1, BH2, BH3, and BH4. These domains correspond to the alpha helicases, which dictate structure and function. The anti-apoptotic members contain all four domains, while the pro-apoptotic members contain at least one domain and usually lack the BH4 domain. BCL2 is an anti-apoptotic protein which the BCL2 family is named after. It was the first BCL2 family member to be discovered and is made up of all four conserved domains. Its BH1, 2, 3, and BH1, BH2, and BH3 domains are in close proximity and create a hydrophobic pocket which can accommodate a BH3 domain of a pro-apoptotic BCL2 family member. The BH4 domain of BCL2 binds to the IP3 receptor, thus giving it its apoptotic properties. The BH4 domain of BCL2 is required for its binding to the IP3 receptor. This binding to the IP3 receptor is essential for the blocking of the apoptotic increase of mitochondrial membrane permeability through direct interaction with the voltage-dependent anion channels. Deletion of the BH4 domain eliminates the pro-survival activity of BCL2 without interfering with the ability of the BCL to bind BH3-only proteins. Glutathione S-transferase pull-down assay is a method used to confirm known interactions and also to confirm interaction sites between two or more proteins. The first step is the binding of the bait protein to the affinity ligand. In this study, glutathione is the affinity ligand and the IP3 receptor is the bait. After binding of the proteins, they are then washed to remove any unbound baits. Next, the prey, which in this study is BH4, are then bound to the bait and washed to remove any unbound preys, or BH4s. After washing, the ligand bait prey complex is then eluted and analyzed using SES page gels. Figure 1a shows the structure of the IP3 receptor. It is made up of a suppressor domain, an IP3 binding domain, a transmembrane domain, a gatekeeping domain, and a regulatory and coupling domain. This regulatory and coupling domain is also known as domain 3, and it is where the BCL interacts with the IP3 receptor. The top part of figure 1b shows us what they put in the GST pull-down assay. While the bottom part is what was pulled down. Delta BH4Cl does not bind to IP3, so it's not pulled down by either the D3 domain or the D6 domain. The BCL2 binds to the D3 domain, so it gets pulled down right here by the D3 domain, but it doesn't get pulled down by the D6 domain because it doesn't bind, because BCL doesn't bind to the D6 domain. As mentioned earlier, the B4 domain is necessary for binding or interactions with the IP3 receptor. In Figure 1C, they use immunoprecipitation to show evidence for this occurrence. So basically, the immunoprecipitated BCL2 with the IP3 receptor, they use two different forms of BCL2, and also they use BCL2 that will lack in the BH4 domain, and they intentionally use a higher amount of those lacking the BH4 domain just to see if they were going to get a weak interaction or not. Here to the right of the figure, you have the results, and we see that the BCL2 is able to bind to the IP3 receptor, but the BCL2 that does not have the BH4 domain could not bind to the IP3 receptor, so we did not get any precipitation. That's why we don't have a band on the gel. In Figure 1D, they use pull-down assays to determine whether BCL2 BH4 domains alone was sufficient to interact with the IP3 receptor. They use a fluorescently labeled fusion protein, TATBH4, to show that GST IP3 receptor domain 3 was pulled down by BH4, but not the GST IP3 receptor domain 6. So right here, this is a histogram showing like the fluorescent counts when they use the fluorescently labeled TATBH4 bound to domain 3. Domain 6, we have a very small interaction, which is not very high, but when you use the domain 3 with the BH4, you can actually see like a huge difference, indicating that domain 4, indicating that BH4 actually binds to domain 3. And to the right, which is figure E, they use co-immunoprecipitation to show the interaction of the full-length IP3 receptor with the fluorescently labeled TATBH4. And we can see that we had precipitation right there when we use the BH4. This shows that BH4 on its own can bind to the IP3 receptor. It doesn't need the help of the BH1, BH2, or BH3 to be able to bind to the IP3 receptor.
To investigate, BH4 peptide inhibits IP3 receptor channel activity. Calcium flux assay was a technique in this experiment, while mouse embryonic fibroblast was a cell type. Different components were loaded in thapscargin, which is selective inhibitor of sarcoendoplasmic reticulum, then and flushed through with ethylene glycol tetrastic acid. Calcium was released from IP3 receptor and found that if BH4 flushed by itself, it's a greater inhibition over the time that it doesn't allow it calcium to release. And this data was taken per 2 minutes. While additional peptide 2, which is a specific inhibitor of BCL2 IP3R interaction, had the least greatest amount of inhibition. Here's in the green one. Fecal by itself was similar rate of a loss as the green one, while control had the greatest fraction a loss over the time. To explore the effect of different concentration of BH4 peptide and control peptide on the IP3-induced calcium release, different concentration of peptide were measured. At this particular concentration, which is the lowest concentration compared to other, we can see more influence of control. An increased concentration of peptide, then percentage of control start decrease. Experiment result was that BH4 domain is sufficient to inhibit IP3 receptor channel opening in vitro.
Here they were investigating if the BH4 domain can inhibit the anti-CD3 induced calcium elevation by BH4 fusion peptide in the WEH172 cell. Part A shows calcium traces of calcium elevation by the anti-CD3 antibody in the WEH172 cells that were treated with the TAT control and the TAT BH4 peptides. TAT-BH4 inhibited the anti-CD3 induced calcium elevation shown in the green. Also shown in part B, the histogram showed TAT-BH4 having the less calcium elevation. They also investigated the PEP2 reversal effect on the anti-CD3 induced calcium elevation. Part C and D showed a histogram of various peptides and calcium elevation peaks. The peptide 2 blocked the inhibiting of the anti-CD3 induced calcium elevation by both the CL2 and TAT-BH4, both shown in red. Here they investigated if the TAT-BH4 had any effect on the calcium elevation induced by the IP3 ester. Part E shows the calcium traces of the calcium elevation by IP3 ester that were treated with the TAT control and the TAT-BH4 peptides. The TAT-BH4 did not inhibit calcium elevation induced by IP3 ester. In part E in the green, it showed no peaks to the calcium, as well as part F in the red box showed low calcium elevation.
They studied the linkage between IP3-induced calcium elevation and apoptosis. They used the Jurkat human T-cell leukemia line, and 4A is a representative of calcium traces recording of the calcium elevation induced by anti-CD3 antibody in the presence of TAT control or TAT-BH4 peptides. They found that TAT-BH4 inhibits the anti-CD3 induced calcium elevation. 4B is a histogram summarizing the results of the average peak of the anti-CD3 induced calcium elevation in response to the different peptides. To determine if the TAT-BH4 has anti-apoptotic action similar to the full-length BCL2, the Jurkat cells were pre-incubated with TAT-BH4 for 1 hour and then treated with anti-CD3 antibody in the continued presence of the TAT-BH4 for 24 hours. The cells were then quantified according to the percentage of the cells displaying apoptotic nuclear morphology. They found that TAT-BH4 inhibits the anti-CD3 induced apoptosis. The red arrow here shows that anti-CD3 with the presence with TAT-BH4, the apoptosis percentage is released compared with only the anti-CD3 by itself or with anti-CD3 with a TAT control. Co-incubation with TAT-BH4 with TAT-PEP2 was used to study the TAT-BH4 anti-apoptotic action. The result is that co-incubation with TAT-PEP2 prevented the inhibition of the anti-CD3-induced apoptosis by TAT-BH4. Thus, PEP2 inhibited the action of BH4. This red arrow shows that anti-CD3 with TAT-BH4 showed reduced apoptosis, which is BH4 is inhibiting the anti-CD3 induced apoptosis. In contrast with the anti-CD3 without BH4 and with PEP2, it shows that PEP2 induces apoptosis. And then to confirm that PEP2 inhibits BH4 action, they used anti-CD3 with TAT-BH4 and TAT-PEP2, and then the apoptosis inhibition was removed.
BH4 peptide binds the regulatory and coupling domain of IP3 receptor and inhibits IP3 dependent channel opening. calcium release from the ER, which is endoplasmic reticulum, and calcium mediate apoptosis. BH4 domain is sufficient to bind IP3 receptor. The major function of BCL2 family is to preserve mitochondrial integrity, thus maintaining ATP synthesis and inhibiting release of a pro-apoptotic factor like cytochrome C, which active caspase cascade. However, mitochondrial membrane integrity is disrupted during apoptosis by two mechanisms. One involves direct permeabilization of the outer mitochondrial membrane by a pro-apoptotic like PAX and PAC. Another mechanism involves induction of mitochondrial permeability transition, mainly response to calcium release from the ER and mitochondrial calcium overload. BH domains 1 to 3 are responsible for forming hydrophobic groups that bind pro-apoptotic relatives, while BH4 is responsible for binding the IP3 receptor, thus inhibiting calcium release from the ER.
Endoplasmic reticulum calcium release. Calcium release can cause endoapoptosis by mitochondrial calcium overloading. Shown in the figure, when apoptotic insult is added in the endoplasmic reticulum lumen, excessive calcium is released from the endoplasmic reticulum, causing calcium overloading in the mitochondria. Resulting in release of the pro-apoptotic factors leads to apoptosis. However, when pH4 is present, the domain inhibits calcium release in the endoplasmic reticulum by the binding of the IP3 receptor, which causes the BCL2 to inhibit any path that can induce apoptosis. This just further confirms that the results previously talked about in the study that the BH4 domain is able to inhibit calcium elevation. Mitochondrial and endoplasmic reticulum crosstalk. Mitochondrial and endoplasmic reticulum crosstalk has a close proximity that plays a role in the cell survival and cell death, which is by the voltage-dependent ion channel, which controls if the mitochondrial calcium elevation will promote cell survival or cell death. The voltage-dependent ammonium channel in IP3 receptor transfers calcium, making the endoplasmic reticulum and the mitochondrial crosstalk strong. However, BCL2 can inhibit mitochondrial calcium overloading and apoptosis by the positioning of the BH4 domain at the endoplasmic reticulum to limit the IP3 channel opening, and another BH4 domain at the mitochondrial to regulate the voltage-dependent ammonium channel. This is a result that the pH4 domain can inhibit apoptosis, as has also been discussed in our results. BH4 domain in its therapeutic values. The purpose of the study was to look at the anti-apoptotic properties of the BH4 domain in the BLC2 and to see if its functions can treat disorders and diseases associated with accelerated apoptosis. As seen in the previous results, the BH4 domain has the ability to inhibit calcium elevation and inhibit apoptosis by inactivation with IP3 receptors. As a result, BH4 domain promotes prolonging cell survival to treat these diseases and disorder. To provide a brief overview of our presentation, Brung and his fellow colleagues used various techniques such as the GST pull-down, the co-amino precipitation, calcium efflux array, and detecting apoptotic nuclear morphology in order to solve and confirm the anti-atopic activities of the BH4 domain of the BCL2 by inhibiting apoptosis and as well as the calcium release from the endoplasmic reticulum by the binding to the regulatory and coupling domain of the IP3 receptor. And this wraps up our presentation. Thank you for listening.